So as an example of how enzymes work, we're going to look at this enzyme here. And this enzyme here is sucrase. And sucrase will break down sucrose into its constituent molecules. And a molecule of sucrose is just ordinary cane sugar that we get everywhere. We also get it from sugar beets, the most common sugar that we use. Table sugar is sucrose. But that's going to be one molecule of glucose and one molecule of fructose. That's what the two molecules are that compose this disaccharide. So there's a monosaccharide glucose unit, there's a monosaccharide fructose unit forming the disaccharide sucrose. Now with random movement, this can fit into the active site of the enzyme like this. So this molecule is now fitted into the active site. And the sucrase enzyme, what it will actually do, it will put a stress on the bond. So the sucrose fits into the sucrase, that's the sucrase, that's the sucrose. And the sucrase enzyme is going to put a stress on this bond here between the glucose component of the sucrose and the fructose component of the sucrose. This is actually called a glucoside bond, it's glucoside bonding. But it's now under stress. So this bond is now under stress. Now, these reactions that break down food products into smaller components are all hydrolysis type reactions. Hydro, water, lysis to break up. So we need to add water to this. So water is going to be added. So strain is going to be put on this glucoside bond by the sucrase enzyme. That's going to allow water, facilitate the addition of water to the molecule. And then what will happen very quickly is that the enzyme will result in the breaking up of the two. And we now have an individual molecule of glucose and an individual molecule of fructose. The digestive process has taken place and they're now free to be absorbed. Physiologically, this would actually happen on the brush border of the small intestine, mostly the ileum, that brush border where a lot of this final stages of digestion take place. So we now have digested two molecules as a result of the enzyme. Now firstly, we notice the enzyme is free. The enzyme is now free to get the next sucrose molecule. The enzyme has not been used up, the enzyme has not been changed. But water's been added, and specifically the glucose has received an OH group, and the fructose has received an H, a hydrogen atom. But that digestive process has now taken place, and the enzyme is free to. They're going to be absorbed, so they'll now be absorbed through the gastric mucosa because they're water soluble, through the villi, into the hepatic portal vein and off to the liver. And the glucose can just go straight through the liver, but the fructose will be converted into glucose the first time it goes through the liver. So the only sugar actually going into the blood will be the glucose. So this enzyme's now free to do the next molecule that comes along as they go off to be absorbed. And the rate of reaction for this kind of process is thousands of substrate molecules, thousands of these substrate molecules per minute. So this is a remarkably fast process going on really quickly. Because if a solution of sucrose is left in a sealed container, it breaks down into glucose and fructose extremely slowly, barely measurable. But in the presence of even a small amount of the enzyme, in this case sucrase, the rate of breakdown is accelerated by millions of times. It's millions of times faster. So just to review a few points. Enzymes are proteins which act as a biological catalyst, facilitating and, as we've seen, greatly increasing the rate of reactions. An enzyme speed up reactions without being used up in the reaction itself. So this was completely free to go on and metabolize or to digest the next substrate molecule. So what's happening is there's a substrate. In the case we looked at, the substrate was sucrose. It was broken down into the product, which was glucose and fructose, and the enzyme facilitating that process was sucrase. But this is the way it works. The substrate is converted into the product, but only under the influence of the enzyme. Now it's interesting to think about how enzymes are named, naming enzymes. And the principle often is it's the substrate or the substance being acted on, and then an A's on the end. So for example, sucrose is digested by sucrase. So the A's indicates that it's an enzyme, sucrase. Now, having said that, there are some examples of enzymes which are named before this convention became well recognised. So it's not a universal rule, but generally enzymes end in A's. So it's the substrate, sucrose in this case, followed by A's. So this was a sucrase enzyme. And enzymes can be classified by the type of chemical reaction they catalyse. So the example we looked at was sucrose going to glucose and fructose by the action of the sucrase and the adding of the water. And we noted that the glucose ended up with an OH and the fructose ended up with an extra H, but they broke down into separate units. So these are both separate units at the end of this process. Monosaccharide units. And so this reaction, as we can see, this reaction here involved adding water. So this reaction involves adding water, a water molecule, to break the chemical bond. And that's why it's a hydrolase. It's a hydrolase enzyme. So these are, all digestive enzymes are actually hydrolase. So hydro is the prefix meaning water, and as we know, Ase means enzyme. So they're hydrolase enzymes, enzymes which break down by the addition of water. And all digestive enzymes are actually hydrolase type enzymes. Now, just before we go on to look specifically at the individual digestive enzymes, I think we'll just notice one more thing about enzymes. There can be anabolic enzymes or catabolic enzymes. So these hydrolase enzymes in the digestive system, are they going to be anabolic or are they going to be catabolic enzymes? Well, the answer is they're going to be catabolic, because a catabolic reaction is breaking large molecules down into smaller molecules, whereas an anabolic reaction is going to build smaller molecules up into a bigger molecule, such as synthesizing a protein. And this is interesting because enzymes can be intracellular or extracellular. So intracellular means the enzyme is working in the cell, whereas the digestive enzymes are going to be extracellular. They're working in or very near the lumen of the gastrointestinal tract, so they're actually working outside of the cells. But many enzymes are intracellular. They are working within the cell, so within a cell here. And in a cell there are untold numbers of complicated chemical pathways going on. So I'm just going to make a really simple one. So sometimes we need product A to be converted to product B, to product C, to product D, for example. And these stages in this biochemical processing are all going to be facilitated typically by different reactions. So that's going to be facilitated by A's, because A is the substrate. So that would be A's there, broken down by A's. This next step will be facilitated by B's, because B is now the substrate, and A's means enzyme. That step will be facilitated by Ca's to get the final product we want, which in this case is D. So these would be intracellular enzymes working within the cell. But in the next video in this series, what we want to do is go on and look at the specific extracellular catabolic enzymes involved in the processes of digestion. That is the process of breaking down complex food molecules, which are typically insoluble, into highly soluble, smaller products of digestion fit for absorption into the bloodstream or the lymphatic system in order to be used as nutrients and providers of energy in the rest of the body.